Hello everyone! In this video, we'll be discussing one of the central cells in adaptive immunity, T cells, and their development from a hematopoietic stem cell to a mature T cell in the thymus. T cells are an essential part of our immune system. It is one of two types of lymphocytes alongside B cells. Specifically, T cells are important for cellular immunity, which means recognition of self cells that present foreign antigens on their MHC molecules, as opposed to B cells and humoral immunity, which means the recognition of intact foreign molecules through antibodies. T cells matures in the thymus. Thus, its name, T, stands for thymus-derived. Let's first talk about the origin and migration of T cells. In a review article written by Ronald Germain from Nature Reviews Immunology, T cells originate from hematopoietic stem cells in the bone marrow, a primary lymphoid organ important in the creation of immune cells. In the bone marrow, hematopoietic stem cells first differentiate into a common lymphoid progenitor that can further develop into either a T cell, B cell, or natural killer cell. However, once there is entry into the thymus, the progenitors can no longer differentiate into other cells, beginning the pathway specific for T cell development. Progenitors that specifically become T cells are referred to as early thymic progenitors. These common lymphoid progenitors leave bone marrow and are released into the bloodstream until they encounter the thymic epithelial cells, at which point the interaction between the two cells can facilitate the entry of common lymphoid progenitors into the thymus. Once in the thymus, the progenitor can rapidly proliferate and follow a step-by-step process to become a fully mature T cell. In the previously mentioned review article, the early thymic progenitors that enter the thymus cortex express neither CD4+ nor CD8+ co-receptors and are thus called double negatives. Double negative cells undergo several stages termed DN1, DN2, DN3, and DN4, with the expression of certain surface molecules such as CD44 and CD25 changes. Finally, the cells start expressing both CD4+ and CD8+ co-receptors, thus becoming a double positive cell. During the DN3 through double positive stages, an important step called TCR rearrangement happens to generate T cell diversity.
Each T cell produces a unique T cell receptor, TCR, that allows it to recognize a specific antigen. However, there are millions of different antigens present in the body at any time, and we need a huge repertoire of different TCRs to be able to efficiently recognize them. A book titled Immune Biology: The Immune System in Health and Disease, written by Jian Wei, described the generation of antigen receptors and the organization of loci to be similar to that of the immunoglobulins in terms of gene segments and both light and heavy chains. There are only 3 genes that code for the TCR protein, called the V, D, and J genes respectively. Therefore, there must be additional mechanisms to generate diversity. The first source of diversity is polymorphism. In humans, there are multiple alleles of each of the V, D, and J gene that can be rearranged into specific VDG combinations. This greatly expands the number of different TCRs possible from a limited pool of genes. The second source of diversity is junctional diversity. When the V, D, and J allele recombine, Artemis can create random nicks at the ends of coding sequences. And TDT can add additional random nucleotides in. This creates asymmetric ends of each allele to be sealed together. Because the cut and the nucleotide addition are both random events and can happen at any position in the sequence ends, it generates additional diversity at the junctions between V, D, and J genes. Many times, these random nicks and nucleotide additions result in a shift of reading frames, which produces non-functional TCRs. In these cases, the cell simply dies. there is a trade-off between efficiency and diversity. If the T cell successfully generated a function TCR, it will be expressed at the surface of the cell, which triggers both positive and negative selection. This process is also described in the review article mentioned earlier, but also in another review article from the same publisher written by Klang et al. Focuses on the positive and negative selection during T cell development, where in order to make sure that these T cells are capable of recognizing our own cells' MHC molecules, thymic cells present self-peptides on their MHC molecules, which conduct survival signals of the T cells by binding at their CD4 or CD8 co-receptors. This process is called positive selection, during which double positive cells become either CD4 or CD8 single positive cells. In addition, to ensure that our T cells does not induce autoimmunity or attack against our own cells, T cells that react strongly with self-MHC complexes are deleted in the thymus during a process known as negative selection. After positive and negative selection, we have a repertoire of mature T cells that can recognize self-MHC molecules but binds to foreign antigens only. These mature T cells will exit from the thymus and patrol the body for potential infections.